Welcome to the Diary of a Medicinal Chemist.
So after the target identification and validation, then we have the lead identification and optimization. Now, it is important to note that most drugs are not discovered. What is more likely discovered are called the lead compound. Now, in modern rational drug design approach, there are a number of characteristics that a lead compound should have before it can be regarded as a lead compound. The first one is that it interacts with the target in a manner consistent with that needed to achieve the desired biological effect. Also, that it is amenable to synthetic modifications to improve, of course, the properties like potency and so on. Also, it possesses and can even be modified to possess properties consistent with that needed to, of course, reach the target after administration via a suitable route. Could be orally, could be intravenous, could be via the buccal cavity, and so on. So there are so many routes of administration of drugs that we know. It could be transdermal, it could be intramuscular, and so many other ones that we know. So it is important that it has these features so that it is amenable to synthetic modifications. Because just as I said, it is possible that although the ability of a lead compound to interact with the target is not enough. Is not enough when it comes to drug discovery to say it is an ultimate drug until we are able to also assess the journey, because the lead compound is not going to fly to the target. It has to go through a number of barriers within the human system. For instance, if it is used orally, it has to go through from the mouth to the gastrointestinal tract to the stomach, then down to the small intestine, to the large intestine, and so on. And a number of things occur, a number of processes occur while it transits through the gastrointestinal tract. And again, it is sometimes a function of the structure of that compound. For instance, when you look at the gastrointestinal tract, it has a temperature gradient in the sense that in the stomach it is acidic. You have a pH ranging from 1 to 2. So if you have an acidic drug going into the stomach, it is not going to be ionized in the stomach because of what is described as the common ion effect. So it's not going to be ionized in the stomach. So it has to go, it is fairly absorbed. It is absorbed better in the stomach for acidic drug. But if it is a basic drug, it will be ionized in the stomach. And once it is ionized, it cannot go through, absorption cannot occur in the stomach. Because when you look at one of the properties, one of the conditions for absorption to occur is that that compound, your compound must not be in the ionized form. Now, having said that, an early researcher by name in Pfizer, one of the leading pharmaceutical company in the world, in I think 2000, around 2001 or 2003, did propose what he described as a rule of five, guiding any drug that is going to be administered as an oral drug, and that is called the Lipinski rule of five. And of course, that for very many years, that rule of thumb was guiding angel, if I may use that word, for a number of pharmaceutical companies in the world are trying to see that the drugs that they develop are tailored towards being in compliance with the Lipinski rule of five. Now, but we have seen that majorly, particularly though for many of us that are into the practice of medicinal chemistry, find out that major, even natural compounds, most of them that are used, you will see them not even obeying the Lipinski rule of five. So it is not, even Lipinski himself did say that it is not sacrosanct, it is not an oxymoron, that if your compound does not comply with the Lipinski rule of five, that it falls within the exclusion list, and you have to be watchful for that. But overall, it is a fairly good guide in selection of your lead compound if you are designing a drug that is going to be an oral drug. So it gave us four rules. He said the first one is that the log P should be less than 5. The hydrogen bond acceptor should be less than 10. The hydrogen bond donor should be less than 5, and the molecular weight should be less than 500, and that is why 500 gram per mole, and that is why it is essentially called the Pinsky rule of 5. It doesn't mean that it has 5 rules, but it has 5. If you look at, these are multiples of 5, and